Our speaker today is Maxime Lucas, who's going to be talking about revealing patterns in brain activity with persistent homology. Over to you.
Thank you, Jason, and thank you for the nice invitation to your seminar. I'm happy to speak today about these results using persistent homology and applied to brain activity. And so it's the first time I speak about them. I've been working on this project for some time.
I'm not sure what happened there, but let's carry on.
No problem. So I work now, I've just moved back. I'm originally from Belgium. I just moved back in Belgium April now. And I'm part of these two departments, mathematics and also a laboratory of mycology, where I do some interdisciplinary work. All right, so let's get started. So I wanted to start with a general introduction and then diving deeper on two kind of projects and showing you two kind of sets of results. And one of the big questions often in biology and in medicine is that you observe behaviors, let's say, in a general way. So this could be some skill level, or it could be some symptoms. How much are you shaking? How much are you feeling bad? Or how much, in this case, will be how much are you susceptible to hypnosis, for example? And then you could ask, in very general terms, can we find signatures or can we understand this behavior or these symptoms from what we observe, for example, in the brain dynamics? Can we observe some causes or at least correlation between what we observe and what's happening in the brain? Okay, so this is kind of general. And so from my background is theoretical physics and dynamical systems. And so in this case, we could view the brain as a big dynamical system. And so in this case, it would be a very high dimensional system made up of many interacting neurons. And so all of these neurons have their respective dynamics and they interact and they create a very complex dynamics. And so the result of this big high dimensional system can be very diverse dynamical regimes. Okay, so for example, I'm sure you all know that when you're asleep, your brain works differently than when you're awake or when you're running or when you're thinking or when you're doing different tasks, for example. Okay, and so you could see these different states, brain states, or these different tasks in dynamical systems vocabulary. You could see them as different dynamical attractors to which the system, the brain, will be attracted under different circumstances and under different stimuli, either internal or external. Okay. And so in theory, if we want to kind of characterize the dynamics, we would want to characterize the associated dynamical attractor. And so that's kind of what we're going to try to do today. So before we can characterize brain dynamics, of course, then again, this is very general, but we need first technology to observe it and record it because this is not obvious, right? And so to do this, there is many now tools, many technologies. For example, we can use electroencephalography. This is a non-invasive technique represented, for example, here. Usually you put a cap on your heads and then you record some electrical signal with different electrodes and different channels at different positions on your scalp. And so this is a non-invasive method that records electrical activity of the brain. Others exist. For example, you could also record the magnetic activity of your brain. You could record with local field potential some invasive techniques in some cases, of course, not on everyone, it depends. So in this case, we will use them on fish in the next part of the talk. But the point is that so all of these observations are partial observation. And what I mean by this is that you're able to observe each of the neurons that compose the system, for example. So what you measure is some kind of observable of the output of the system, and you measure some kind of aggregation of electrical activity in some cases, okay? So this is what I mean by partial observations. And so then you would want to have methods to analyze these recordings, and traditional methods include, so you can work directly on the time series using signal processing, information theory, time frequency analysis. recurrence analysis. There's many different techniques that are well known and well tested in these cases. And one important tool, if you don't want to work directly with the time series, is also time delay embedding. So for those of you who might not be familiar with what time delay embedding is, the idea is exactly that. So the brain is the whole system, and what we measure is only some of the observable of the system. We don't measure all of the neurons. But so by using this time delay embedding technique, there is a theorem about that from Takens. The idea is that you can reconstruct the full system, the full attractor, based on some only partial observations. So we have some multi-channel time series, and we can reconstruct hopefully the real or close to the real dynamical attractor. And that will be very useful, because then you can apply again some of these techniques on not directly the time series, but the reconstructed attractor. So the general pipeline that I will be using in the results I'm going to present to you today is the following. Okay, so we have some raw data that we measure from brain activity. Here I wrote EEG, it could be LFP, it could be other measurements of electrical activity of the brain. So first, either you have some kind of multi-channel EEG, it could be also univariate or univariate time series, depending on how many electrodes you have to measure the signal. Then you do some kind of pre-processing, and then you kind of want to use different representations. So I told you about this first one here, time delay embedding. So in this case, what you do is you use delay coordinates in time, and then you kind of reconstruct this dynamical attractor in some higher dimensional space. So you start from this, and then you get something like this, okay? Another one would be, especially in brain signals, people use often functional connectivity. What is that? That is basically measuring the correlation between different electrodes. So here we have different electrodes, and you could pair by pair look at the correlations between any two, and then you get a matrix, or you can visualize it also as a weighted graph, basically. And that tells you how different electrodes, the signal of different electrode is correlated. Yes, how it's correlated. So in a sense, this one is more spatial, okay? This functional connectivity. In the other case, it's more temporal, because we use this time delay embedding, and we use the information at different times in points in time. Then what we will use, and that's how we arrive then to topology and persistent homology, for those of you who know it. So once we have these, either the functional connectivity representation or the time delay embedding representation, we can use persistent homology. How does that work? Let's take the case, for example, of exactly this time delay embedding. What we can do is we want to look at the topology of this attractor, okay? Here it looks like a donut, basically a torus. And to do that, we can do this at several scales. So first, we want to build a simplicial complex. What is that, and how do we do this? We look at all of the different points, this point of clouds, okay? And we grow bubbles around them with a certain radius. And then we connect these points if they are closer than this radius, basically, if there's some intersection between these bubbles, okay? When we have a triple intersection, then we add a triangle, for example. So this is a generalization of pairwise networks or pairwise graphs. The details are not fully important for us today, but basically you do this with different radii, okay? So you do this for different scales, and then you get what we call a persistent diagram. I will show you in a bit more details in the next slides. So, but with this persistent diagram, you have different points that basically tell you about topological features of this object that we're looking at now, and in particular of this attractor, and it's telling you how persistent they are over different time scales. So you have their radius of birth and their radius of death, and it's telling you if some of them persist among many different R scales or not. From this, I can basically extract the different single number metrics, scalar metrics, and so I want to summarize this more complicated object by simply like 3 scalars, basically a vector. And then I can compare for example, I do this for one fish, okay? And then I can compare it to the recordings of another fish by comparing the vectors. And by doing this, I can compare their topological metrics, which is to say the metrics that measure some topological metric of the reconstructed dynamical attractor, okay? So this will be the general pipeline, and I will tell you more in the next slides. So I have recordings, I represent them by some kind of embedding, and then I apply persistent homology and look for topological metrics, which I can then compare between subjects, conditions, etc. That's the idea. So a few more words for those of you who are not familiar about time delay embedding. What is it? So we start from a full, as I was saying a minute ago, we start from a full system, for example, that we know of. For those of you who know the classical example of the Lorentz attractor, for example, so you have three variable in this case. You could think that could be a brain with 3 neurons, say, and then you observe only one of them, you get a time series, and then you want to try to reconstruct this by using some method of time delay embedding. So you use a time delay, and then from one of the 3 variables, you kind of reconstruct the other 3. That's the idea. And then we want to study this reconstructed attractor that hopefully looks enough like the real one to be able to say something about the real one. That's the idea. A visual representation about persistent homology and what I was telling you about this different radii. Okay, so you create bubbles around these points of different radii, and then you get different simplicial complexes. Okay, and then here you can say count, for example, in H nodes, you have different homological dimensions. This is a bit of vocabulary, and you just need to remember. So we look at 2 different dimensions. Here, H0 counts the number of connected components in the simplicial complex. So you start with basically the number of points, and then here you connect 2, so and 2 and 2. So the number of components is a bit smaller, and at the end, you end up with one big giant component. And so there is only one component in H0 that lasts until the end. Okay, so the number of orange, for example, bars here corresponds at the beginning to the number of orange dots that we have for this given R. H0 counts the number of connected components. H1 counts the number of cycles in this structure. So here you see we have 0 cycles. So at the beginning, there's nothing happening here. And at some point, when we start connecting more points, you see that we get what's, I'm not going to define it formally, but you see that there's this closed cycle here in the structure. And so it starts here and it lasts until here, because afterwards for larger R, it disappears. There's no more cycle. So we will look at connected components, H0, and we will look at cycles, H1, of the dynamical attractors. Then, so this is a barcode representation, which is absolutely equivalent to this persistent diagram representation that we will use. The only difference is that now, instead of bars, I'm plotting points just with the coordinates of their birth and their death. And this is orange dots are H0 and blue dots are basically H1. So if you have some points that are further from the diagonal, they have long persistence because they last long. These correspond to long bars in the barcodes in the previous slides. Points that are closer to the diagonal have very short persistent, very short lives. And so they're not very persistent across scale. So usually they're more considered like noise. Okay. From this, I extract 3 topological metrics that I was telling you. The first one is going to be total persistence. The persistence of a point is its distance to the diagonal. So the total persistence is basically the sum of all the persistencies that we can see in this diagram. Persistent entropy is going to tell us about the diversity, how different these persistencies are, because I have a distribution of persistencies for different points. And the Betti number is, in this case, a generalization of the one in mathematical topology, and is just the number of points that we have here. So I will use these 3 metrics to summarize this persistent diagram, and I will use them to compare different conditions in my data. So now I'm going to start immediately by telling you a bit about epilepsy in zebrafish animal models. So epilepsy is basically affecting 50 million people worldwide, which is a lot. It's one of the most common neurological disorders, and it's characterized by recurring seizures that disrupt normal activity, okay? And so, of course, it's a very complex disease, very complex disorder, and there's a lot of interdisciplinary research going on with genetics, processing, machine learning, modeling. Many different types of disciplines are working on this. And there's been a lot of progress, for example, on prediction and detection of seizures. So on a short time scale, right? So that, for example, if you wear devices now that can record your brain activity 24/7, we can be alerted when a seizure starts, let's say, because it's detected automatically. Now, what is a bit more challenging is understanding the mechanism and the causes of epileptogenesis. So this is the development of this disorder before you've had any seizure. Why do you develop epilepsy? Okay. And this is happening, of course, on longer timescales. And we know also from genetics that there is a lot of, but not only because, so there's a lot of different causes and a lot of different types of epilepsy. There's actually a whole classification of epilepsies. And many of the causes are actually unknown. And so we were wondering if we could find signatures of susceptibility to epilepsy, basically in background activity, so in normal brain activity before you develop seizures. Of course, I think the goal of this is pretty obvious, and it could be quite helpful. So the data set that we have is quite good because a few studies, not many, maybe 3, 4, have tried to use TDA for epilepsy. TDA is topological analysis, of course, but in many cases, they use data sets from human cases, and there's not so much control about different genetic mutations, different control over the experiment, basically. And so zebrafish have been used as an animal model more and more in recent years for studying epilepsy, exactly because they have you can produce many. Their lifetime is not so long, and then you can control different genetic mutations, etc., etc. So I don't do the experiments; biologists do them, but this is an in-house data set, and so it's quite nice to have this data basically to be able to try and answer some of these questions. So here what we have is a data set with 2 different fish lines. These two here, this should be an A, okay, ASH1L. Each of the fish line has can have 2 different conditions, either wild types, which is basically the natural version, let's say, and another version with some mutation that is chosen usually by knocking out one gene. These are the names of the genes that are knocked out, basically. And then we look at 2 different types of recordings, basically either during seizure or during background activity. And these are the numbers that we have for all of these cases. You'll see that we have a 5th subgroup. This subgroup is basically the same as this one wild type one with this one fish line, but it's a subgroup of fish that have not had any seizures. So in the other case, this wild type are fish that have had some seizures, and these ones have not. Okay, and then you will see that we will try to compare these. Okay. So again, this is a slight variation of the pipeline that I showed you before that was general, but basically here we have LFP, so local field potential. We put a needle, well, they put a needle in the brain of the fish. And so it's only one electrode, and you get a single univariate time series for each fish. And so we have them for different fish line and everything that I told you before. And so basically then what happens is that we have some pre-processing, including some machine learning algorithm to label the different parts of the recording, right? Because, so for example, you start recording and then a seizure happens and then you keep recording. And so you want first to segment. So being able to say, this is where the seizure starts and this is where it stops, so that you can cut it and then have different recordings with that are separate. And then you want to be able exactly to label, this is a seizure, this is background, okay? Then what we do is, because we only have one single electrode, we cannot do any functional connectivity embedding because there's no space here, there's one electrode. So what we do is we reconstruct the dynamical attractor, like I was telling you. And then same, we apply topological data analysis and we look at these different metrics, okay?
Well, quick question. So you're measuring a group of neurons, right? With the LFP recording. It's not a single neuron, right? It's a group of neurons.
Absolutely. So
And they put a needle, so it's a bit
yeah, absolutely. So the fish have absolutely more than one neurons. I could not tell you how many neurons they have, but definitely more than one. But there is physical space to put only one electrode. So we have exactly one recording that measures the result of, well, multiple neurons. Yes.
which are always somehow the same type, or in each recording there could be different types, or
So yeah, in general,
what
they
makes
try to make it as uniform and consistent as possible. So I know also there was sometimes some difficulties with putting it exactly in the same place, close to the neurons, because depending on exactly where you put it, the signal may just differ. So, but they're professionals, so yeah.
Okay, thanks.
But I haven't done the experiments myself. Yes. But yeah, you need, it requires experience and to be careful. Okay. Any other questions?
I guess to follow up, so is this electrode like supposed to be representative of the whole brain, or like it's just like us, like a small part of the brain? That's, yeah.
So that's also a good question. I think what they were seeing is that if you put it in different places, but I hope I'm not saying wrong things here, but it's supposed to be representative of the whole brain, or at least of the activity of these seizures and non-seizures. Yes, it's not put in a specific part of the brain. I think that's supposed to be different from other parts of the brain. They basically positioned it to have a signal that was as clear as possible.
Okay, thank you.
Sure.
Maybe I can just comment on that. Do you hear me?
Yes.
Yeah, usually LFP is like around 1,000 of neurons that you
Okay.
record. And this basically the electrical field created by these neurons.
Yeah,
Yeah,
exactly.
it's just if it's a needle, it's not the same one that we use in bigger animals, right? So I was curious what is actually happening. But anyhow, it's representative of the whole brain, and I think with this we understand.
Okay. Yeah. I can, of course, check some of these details and yeah, but this is the idea. Okay, so I will go on. And to give you an idea visually, if I compute the persistent diagrams of, for example, a seizure event and a background event, this is what they will look like. Okay, so in black, I have H nodes, so the connected components. In green, I have the cycles. And you can see already that here I have many more points that are, well, first with bigger radii, okay, in green than in here, and also further away from the diagonal. Here I'm showing them on the same scale. I could also zoom in and show you each on their relative scales. But again, if we look at this one from closer, it's still very close to the diagonal and less points further away from the diagonal. So this will be reflected in the measures, the summary metrics that we will see. Okay, so the idea is to have of course, some statistical analysis, and I performed permutation ANOVA with 3 factors, right? So I look at, let's say, one topological metric, one of the ones, the 3 I showed you, and I wanted to see if it's affected by 3 factors: fishline, that has two different values possible, conditions, so that would be either mutant or wild type, and then event, so that would be seizure or background. And then I also included potentially their interactions. And so we wonder, do these factors have a significant effect on the value of the topological metric or not? And what we saw was that, yes, they do. And then, so I will not share all of the details of the statistical analysis, but we focused on 3 main aspects of this. So the first one is that, as kind of is expected, but it's a good validation, these topological metrics are able to discriminate between seizure and background, and you could almost see it already by eye in the 2 examples I showed you. So this was a significant effect with a very large effect size. So the difference between the topological metrics are quite high. For example, here, if you look at persistent entropy, if we just look at this panel here, we compare seizure in green and backgrounds in purple. This is for H0, so H node, so for connected components. And you can see that with persistent entropy, you can already differentiate between background and seizure. This is the case with most topological metrics in most dimensions, so H node, H1. Only here, total persistent, for example, in H node was not able to differentiate this. I'm showing you this result for the subgroup of mutants of the ASH1L fish line, okay, one of the fish lines. If I show you these plots for the other conditions, mutations, and the other fish line, it's going to be very similar, okay? So that's a good check that already the topology of this reconstructed attractor is able to discriminate between these types of events. Then we were wondering, can we discriminate between fish line? Are they going to be different or not? Okay. And so in this case, I looked only, for example, so in most cases, it cannot, okay? So for example, here I'm showing the subgroup of seizures recording for H1, the homological dimension 1 of cycles, and I'm comparing the two fish lines, okay? So blue is one of the fish line, orange is the other fish line. And I'm looking at, for example, mutants and here wild type, and you can see that for any of the three metrics, basically the difference between the two fish line is always not significant. Here it's slightly significant. There's something happening, but most of the time it's really not significant. And this kind of suggests to us that there is at least some shared topology between these events in the different fish lines, okay, in their dynamics. So that can be interesting. But then we were still able, it's not as easy as that because we were still able to see something if we looked at the cycles, okay, H1, and we looked, so it's a very specific case, right? So if we look at H1 and we look at background activity, okay, then we're able to discriminate between the fish lines. Okay, so here, for example, persistent entropy or even total persistence here is able to differentiate between the two fish lines in mutants, but also in wild types. And so first thing that we have to say is this difference is significant, but as you can see, the difference is not as big. The effect size is clearly not as big as it was between background and seizure. So the effect size is much smaller, but still there's something significant. So this seems to say that if we look in the right place and we dig a bit, we can find some differences between fish lines, but in general, it's harder to see them. Sorry, my throat is starting to be dry. Okay, so that was nice. And then we had the last result, which was basically what we were trying to see from the beginning. And we were wondering about, now we saw that in the background activity, we could differentiate between some of the fish lines, but what if we looked at, so only background activity, we fix everything. So we look at one fish line with a single genetic condition, so only wild types, and a single event, so background. And we were wondering, can we distinguish between fish that have had seizures and fish that have never had seizures? And these are the two groups I was telling you about. So we will call the ones that have had seizures wild types, I mean WT, and the one that have not had seizures, seizure-free wild type, okay, SFWT. And interestingly, we could, okay, so if you look here between these two subgroups, we can see differences for all of the topological metrics, okay? And this is kind of interesting because so the only thing that differentiates these fish is that they have had seizures or not. So in a sense, to us, this was suggesting that there is a topological signature of epileptic susceptibility, right? Because so we see that there's this difference just because they've had or not seizures. And so here we're having two hypotheses, right? So either the topology was altered by having the seizures, okay? Or it could also be that the topology was altered between having the seizures just because it was prone to having seizures and then it had seizures. Of course, with our data, we cannot differentiate between these two hypotheses, right? But so in either case, I mean, in both cases, it's interesting. And it would be nice to maybe with some further experiments being able to differentiate between these two hypotheses. Okay, so in this case, these are the take-homes. We used a controlled data set on animal models, and we applied a combination of time delay embedding and TDA. Okay, this has been used in other cases we know it can differentiate between different types of dynamics, especially from the simpler models. But so it was nice to see it in use and being able first to discriminate between background and seizure, and sometimes between fishline under very specific lens. And then finally, seeing that maybe there's some topological signature that we can see between from fish that are seizure prone. Okay, I was hoping to have this on arXiv today to share a link with you. Unfortunately, I finished the submission yesterday at 8:02 instead of 7:59, and so it's going to appear on the arXiv tomorrow. I was 2.5 minutes late. But here's the title, and you can see my other collaborators that worked on this project. So now I will finish with another project that was on hypnosis, and the analysis pipeline is very similar. But so basically, hypnosis, there's a lot of actually people studying hypnosis in research contexts, and it's an altered state of consciousness, and people study it not only to study it per se, but also to try and investigate mechanisms of cognitive control, expectation, prediction, and other cognitive aspects. Okay, so one interesting thing is that different people can have different hypnotic susceptibility. Are you more susceptible and easier hypnotized, let's say, or not? And this can be measured by a standard scale, for example, here, the Stanford Hypnotic Susceptibility Scale. And what we mean here by hypnosis is not what you would see in shows or at the circus or things like this. Usually, it can be used, for example, also to help, similar to anesthesia in before or after surgery. It's conversational hypnosis usually. So this scale, you're scored as lowly susceptible to hypnosis if you have a score less than 3, and highly susceptible to hypnosis if your score on this standard scale is higher than 9. And there's many studies that show that these susceptibility correlates with other metrics. For example, here's a reference to a paper where they show that depending on your susceptibility will display different functional equivalence between imagery and perception or action. So what this means is that they had this experiment where they had people imagine an action, okay? So for example, raise your arm and something like this. And then they compared this to when they were actually doing the action rather than just imagining it. And in the case of highly susceptible people, what they saw in the brain was much more similar in both cases. So imagine and acting than in the others. So there's a lot of interesting correlations between susceptibility and other aspects of what's happening. And so the questions that the collaborators they had the data, and they were wondering, can highs, so we will call them highs, so the highly susceptible highs and lows, can they be differentiated by their EEG activity following hypnotic induction? So can we see the difference between high and lows after hypnosis from basically using TDA? Okay. And so what we had from them is a data set again, this time not with LFP, but EEG. So with many more electrodes in this case. So there's a cap like this. I might be wrong. I think it was 64 electrodes. I had a funny story, very brief parenthesis, but all of this now we rely a lot, I think, at least many of us, on Overleaf, and Overleaf is not working since this morning at 11. So I had trouble retrieving some of the things I wanted to show you, but it's fine. I have most of them, including, for example, the number of electrodes. But yeah. So for each recording, we had the experimental procedure was such that they have recording before, during, and after the hypnotic induction. And there's also an event after the hypnotic induction, which is called they're having a hypnotic dream. So we have these recordings of EEG at these 4 different moments, and we also have the hypnotic susceptibility scores of each of the patients and some other what's called PCI score. It's a self-assessment experience telling us about their internal subjective experience of the experiment, basically. And so that is what we have. And again, I'm showing you the same picture just as a reminder, so that's what we're going to do. And in this case, we're not only going to use time delay embedding, we're going to compare time delay embedding with also functional connectivity, so some kind of spatial embedding, and then apply the same pipeline and use topological analysis. And I'll just show you the 2 main results. Basically, what we saw was that here we looked at first each of the topological metrics separately, like before. So how does persistent topology change and everything. But also we could compare vectors, right? How does this topological vector change between different conditions, okay? And so in this case, we notice, for example, that the highs have a lower persistent entropy, okay, than the highs. So here the highs in blue have a lower persistent entropy than the highs. This here I'm showing homological dimension 0, and at the bottom homological dimension 1. And what we also saw that was quite striking is that you see here the blue bars are always higher, meaning that between this is showing you how much their topology changes between 2 conditions. So how much do the high change their topology between before and during the hypnotic induction? And you see that the blue bars are always higher, showing that the topology of the highs is always consistently changing more than that of the lows, okay? When we look at the temporal embedding, time delay embedding. And we saw kind of the opposite when we looked instead at the functional connectivity, so the spatial embedding. In this case, you see that the blue bars are mainly lower than the orange bars, showing that they are changing less topologically. So the highs are changing less topologically than the lows between different conditions. And same here. In this case, we have the opposite trend. So persistent entropy of the highs is higher than that of the lows in this case. So it's quite interesting that we see opposite patterns and opposite trends depending on whether first you're high or you're low, and second, depending on how we do the embedding. If we look more at what's happening in a sense in space or what's happening more in time about your dynamics. And so that's kind of the comes of this one. And we also tried to make a small model that I cannot show you here because the image is on overleaf, but the idea was that we found some pattern. If you try to generate data, so instead of looking at functional connectivity or actual data, if you try to just generate a distribution of points in 2 dimensions, say, and you make it more uniform, or if you make it more clustered, okay, just randomly, and then you apply topological data analysis and you see what's happening, we notice some of these patterns, some of these same patterns. So I cannot tell you which one was which in the end, but in the sense that if you look at space, it looks like the highs have a more clustered, say, dynamical attractor, but in time, the other ones have a more uniformly distributed. So of course, what would be nice would be to be able to interpret this a bit more physiologically and see if we can make some checks about what this really means for practitioners. Okay, so that's my conclusion now. And so basically I showed you 2 studies with examples of how TDA can be a powerful tool to analyze brain activity, and especially to try and correlate so the changes in topology with different biological factors. Okay, and that's the interesting bit, when we can correlate what's happening with different biological factors or conditions. So I was lucky to have these collaborators that had data sets with control either genetic mutations or different patients. And yeah, So that's basically it. In fish, the main result would be that we found some differences between the fish that had and did not have seizures, and in human hypnosis that this topology was affected differently depending on the embedding and depending on your susceptibility to hypnosis. That's it. These are the next steps. I basically told you already what I wanted for the next steps in fish. Hopefully we can select one of the two hypotheses that we had for explaining this last result and see if it can be maybe used for epileptic recordings in other species and see what we can see there. In humans, hypnosis would be nice to interpret a bit better these results, not just in terms of theoretical models, but maybe see if we can find out a bit more. And yeah, that's it. So I also have to mention my collaborators for the second study that I realize are not there, but basically it's Giovanni Petri from Sentai in Turin and now the Northeastern University in London, Ruxandra Tivandra, who is working at EPFL and she took all of the measurements and all of the recordings, and Jamie Glimison that's also working on hypnosis and is a more senior author. And that's it. I'm happy to take any questions.
Great, thank you. If everyone would like to unmute and clap or show your appreciation how you see fit. Great. Thank you, Maxime. That was a great talk. I really enjoyed it. Does anyone have any questions?
I have a quick one. Just wondering if you're looking at persistence at anything more than H0, H1, like H2, H3, H4.
So that's a classic question and a good one. So the higher you want to look, so it would be interesting to look. It's quite demanding computationally, so it's not always doable, especially when you have longer time series and that your attractor is in higher dimensions. That's why you will see that most papers that look at these kind of, at these kind of things in data, they go for H0 and H1. I would be interested in seeing H2, H3. Depends. In some cases, I think it's going to bring more information, like, you know, we saw in a specific case with fish. In other cases, we probably won't see anything more.
Chen Yin, you had a question?
Hi, thank you for the talk. So you went from fish, which gives you one-dimensional time series, to human, which gives you like 60-something dimensional time series. Do you encounter problems of like cursive dimensionality when you goes from like one dimension to a very high dimension?
So mostly what I encountered was, as I was saying, sometimes you're restricted by computational resources, and so one of the things also so when you do the embedding, time delay embedding, and you have one time series, it's first easier to estimate the parameters for this embedding. So for example, the embedding dimension and all of this. And yeah, so mostly yeah, computational restrictions.
Dave, you had a question?
Yes, I do. My first question, you actually began to address it. How do you determine the embedding dimension? Did you look at different dimensions? Did you choose an optimal dimension, say for fish, and then similarly for humans? And then I have a second question.
Yeah, thanks for the question. Absolutely. So I didn't go into full details about this, but yes, so there are standards. Basically, in the time delay embedding, there's two parameters. One of them is what exactly the time delay that you're using,
Yes.
so a tau, and then you have the embedding dimension. I use standard techniques, so usually you determine first, if I remember correctly, I think first the tau and then the d. With the standard about closest nearest neighbors. And so basically, yes, I check different dimensions. There are standard techniques to try to optimally determine them. And then I also basically, after I add an estimation, I varied the parameters to see if it was not, the results were stable enough. And in this case, luckily they were. But yes, you're right that selecting these values can be tricky. And I saw now that very recently, I think in the last few years, there's also new techniques that instead of trying to determine one parameter and then the other tries to determine the pair of parameters together. Yeah, so I would be interested also in seeing how, I mean, if it can help us with this, but I checked and it looked stable, yes.
Okay. And then the second question is, for the human experiments, you have 64 electrodes. Are you using all 64 signals or a subset of the signals?
I kept it simple and I used all 64 because otherwise I would have had to choose and to have a criterion on how to select the subsets. So yeah, I did select all of them. I know that there is one study where they kind of tried to look into using TDA also for similar things, but more for detection, and they were interested in seeing where, for example, the seizure was originating. And so first they selected all of the electrodes, and then to try to locate it, they selected one by one. So they looked at each one of the electrodes and redid the embedding, and then with some of the electrodes, they noticed that there was some change, and they noticed the seizure starting, and in others not. So they were able to kind of pinpoint where this was happening.
Okay, good. And that raises, so with the human subject experiment, it wasn't epilepsy, it was this hypnosis state.
Yes.
And you mentioned the ability to, the brain reacting to imagining the motion
Yes.
as opposed to doing the motion. And does this involve mirror neurons?
Probably. This is a bit too far from my area of expertise, but I think so. And hopefully I don't think there's any study actually stating that in this context, but it would be interesting to have somebody look more into this. Yes.
Yes. Yeah, the reason I asked that is mirror neurons come up in ASD, and they're located in certain regions of the brain are more, implicated in that.
Mm-hmm.
So if you were to select a subset of the time series reflecting regions of the brain where you expect certain types of responses,
Yes.
that might be interesting.
Yeah, that's a very nice suggestion. Actually, if you have any suggestion or anything any ideas you have, maybe we could talk and check. I know there's another project also where we started checking subsets of neurons also based on different, so for example, frontoparietal or different areas, because the practitioners expected maybe to see something different. But yeah,
Right.
if you have any suggestion, happy to try.
Let me do a little research and I will get back to you.
Sure.
Thank you.
Thank you.
Arne, can I go?
Yeah, and related to Dave's second question about number of electrodes. So I work with high density EEG, 128 channels, as well as functional and structural MRI. So now we're talking tens of thousands, if not 100,000 possible voxels.
Yeah.
Is there any fundamental limitations to this technique in use of many different channels? Of course, you could do region of interest. You could do all kinds of different ways of subsampling that space. But is there any fundamental limit to doing so?
I don't think there is. So the only, well, it's not fundamental, but it's still an annoying limit is the computational resources, because if you get more data and more channels and all of this, then it's going to be, it's going to ask you for, probably then you want to reconstruct a higher dimensional space, and it's going to be very costly in terms of memory and computation time. So that would be, I mean, one would need to try and see if it's doable in a finite amount of time, basically.
Is there a way, like in subgraphs, to be able to look at simplicial complexes in parallel, meaning to parallelize as some subgroups of these channels?
Probably, there's probably ways to optimize this, and I know that, so yeah, there's probably ways to optimize it. So it would be worth giving it a shot and trying to see what we can find. Yeah, because I mean, having such a data set with good resolution would be very interesting. And then one would, if it works, could be even interesting to then try to see the effect of the number of channel, right? So you could try to do the same, sorry, and then say with the full data set and then by coarse graining at different levels and see how it changes basically. How it
Great,
changes
thank you.
the output. Yeah, it
Thank
would be
you.
interesting.
Yeah.
Thank you.
I have a question actually. So the zebrafish one where you had kind of the 6 plots for the H1 and the H0, and there was one where it wasn't statistically significant, the difference. I don't remember which one it was. And then the other
This
5
one?
were.
Maybe
No, the
this
one where
one?
kind of one of them wasn't statistically significant. The top left wasn't, and all the other 5 were.
Yeah, this one.
Yeah, this one. So what does that mean? So the total persistence is the same, but the persistent entropy is varying quite a lot. So in terms of kind of the persistence, what's happening here is you've got like so the total persistence is the distance from the diagonal. Is that the one?
Yeah, exactly.
So that's kind of the same, but you're having the other ones are varying quite a lot. I'm wondering if you kind of, is there anything you can deduce from this that kind of tells you about how the persistence is behaving?
Yeah, that's an interesting one, and that's what I was trying to do also with this simple kind of model I was telling you about, generating more uniform or more clustered data. It's not always immediate to find a configuration that can fit these different metrics at the same time. So in this case, you have very much more a higher persistent Betty number in the purple one, right? So that would mean you have many more points if yeah, here, whoops, many more points orange than in the other case. So then here it's telling us, so we have more diversity in the purple one than in the other one, but then if you look at the total persistence, somehow it's comparable. So yeah, the way I approach this is by looking at the different metrics and the different interpretations. It's hard to get a full picture other than just describing each of these separately. So in this case, it's more variable. We have more, but the total length is similar. So yeah.
So Jason and Maxime, is it possible that I'm thinking more from a functional connectivity standpoint, which I know this is not, but if it were, then it seems to me that the total persistence, which is the sum from the diagonal of all the different channels, if you have the total persistence doesn't have a significant difference between these two, then it suggests to me that there's just a baseline of functional connectivity or baseline of persistence across the different radii for the individual points in that high dimensional space. It seems to me that just when you're talking about brains, you're not interested, like in a seizure, sure, you're interested in that there's a gross kind of disruption in pattern of brain activity or level brain activity, but almost invariably you're interested in variability, like what is active, what is not active. And the part that I think topology plays a real role is in saying as much as inactive as active for like holes in different dimensions. So the persistent entropy, I think naturally, and the Betty numbers seem far more interesting to me than the total persistence, because if everything can persist, that doesn't really tell you much about the variegated brain activity. There's not much of a pattern to be seen. That would be my guess.
Yeah. And yeah, no, I agree. And in other papers, I also saw that persistent entropy usually was a better discriminator. I completely agree. Thanks a lot for the comments. Also in this case, because total persistence, in a sense, total persistent is telling you in total how far away from the diagonal you are. And so in a sense, how much topological signal you have, because you can interpret points close to the diagonal as being more noisy because they're not very persistent. And so in this case, it's kind of, as you were saying, giving you a baseline that's the total topological signal in this system is similar. And then what does change is, for example, with persistent entropy, the distribution of these persistencies and then the number of these features. So I agree. I completely agree. It's a good one. In this case, having them, it's telling you kind of giving a kind of a baseline about the whole complexity of the topology that's happening here. Yeah, thanks.
Yeah, if there were an inflection point where phase locking or something where you can imagine that all of a sudden everything would be connected in a sense, then that's I don't know if that's interesting for seizures, but it's generally less interesting than seeing I would say different levels of persistence in different channels.
Yeah.
Across different channels, yeah.
Yeah, absolutely. I agree.
Nice. Yeah.
Also, I'd be really interested in the hypnosis if this were done from region of interest analysis. I know this was not done with functional MRI, and so you're really just trapped at like EEG, which is very coarse and crude, very great for temporal, but very poor spatial.
Yeah.
It'd be really great to be able to look at things like the default mode network and things like that for suggestibility for hypnosis, because then you can imagine looking at particular networks and seeing how they behave versus other networks behave.
I agree. So yeah. And especially because you were asking about larger data sets in some sense. So if we do functional connectivity, what is very costly is the time delay embedding. Functional connectivity is not very costly. So yeah, it's much easier. Then when you apply TDA, it might, depending on the side of your functional connectivity, it might be a bit longer, but I think it sounds much more feasible with functional connectivity than with time delay embedding. And then I agree. So there's what you were mentioning with different regions of interest and see how different default networks behave. So it's not exactly the same kind of method, but with collaborators, we've had a paper come out very recently that, among other things, so it's looking into the topology and building simple complexes from time series again of brain data, and looking at what's happening between different default networks. So if you're interested, I can send you the link definitely.
Nice. Any more questions?
I actually have one more question in this direction, because now got really interested in this. So this is for the time delay embedding of the fish, right? This figure that's here.
Yes, absolutely.
So I wonder if when you do the time delay embedding, if you're somehow normalizing the point cloud to be more or less in the same range in space or something like this, because having the same total H0, wouldn't that mean that generically, if you look at the point cloud of your time delay embedding, which you can think of kind of sampling your dynamical system, then there is a point in which everything becomes connected once you draw an edge that it's at maximum this distance. This is somehow like the total distance thing. Well, saying that the entropy of one is fixed up and the entropy of the other is fixed in another means, one of them, the components are kind of blending together at the same time, while in the other, they're blending at different times, and you have components of different sizes in this like point cloud of that's sampling your dynamical system. So,
yeah.
But in this figure, I thought you're not normalizing because the guy on the right looks very squished in the left. So I'm a bit confused about it.
No, absolutely. So thanks for the question. So far, I'm not normalizing. There's pros and cons about normalizing, basically. Now that I've done the analysis without normalizing and looking at actually what's happening, it would be nice maybe to see something complementary by normalizing, basically, and comparing at when we put them on the same scale. Yeah. In a sense, mostly this difference of scaling comes from the difference between background and seizure. And in a sense, by doing this statistical analysis, we already saw other factors, sometimes like fishline, having an impact on these topological metrics. So in a sense, we can see we can see beyond this difference in scale in that way too, but we could probably see even more by normalizing and getting rid of that dimension in a sense. For us, it was already good to see this big difference between background and seizure just from the difference in scale in a sense, because then somehow it validates it. But yeah, thanks.
Okay, I will stop the recording now, and then you can have some more questions if anyone wants to afterwards.
Thank you.